Good morning, everyone. I welcome everyone for this Cognitive and Neurosciences webinar 2022. I thank organizer for inviting me to give a guest lecture on my research work titled Is the Gut Microbiota a Neglected Aspect of Brain Disorders? I myself, Dr. Yuvaraj, Assistant Professor in AIMS, Hyderabad. Gut microbiota. Bacterial ecosystem is diverse with multiple bacteria having it from pathogenic to non-pathogenic. It is one of the main member of adaptive immune system. It protects against pathogens. There are various external stimuli that can modify our gut microbiota, like physical activity, stress, diet, and drugs can modulate the microbial environment. When the microbial environment, the normal commensals of the gut bacteria, it helps to protect from pathogens by interacting with pathogen recognition receptor. It acts as an anti-inflammatory because of short chain fatty acid release from the dietary fibers. It has immune homeostasis it can maintain by causing a Treg cell expansion. On the other hand, a dysbiosed gut. Dysbiosis because of our lifestyle changes, the diet modification like high fat and low fiber diet, drugs like metformin can cause dysbiosis. This dysbiosis means alteration in the gut microbial flora. This dysbiosis can cause autoimmunity by increasing the inflammatory signals. By producing long chain fatty acid, it can cause inflammation, and by the way of biofilm formation, it can change the tight junction present in the bacterial gut microbiota so that pathogen can escape into the circulation. In autism spectrum disorder, pathogenic bacteria will shed pathogen associated molecular protein, that is PAMs, which can be recognized by pathogen recognition receptor PRR. Upon recognition by PRR, can cause the activation of nuclear factor NK, MAP kinase MAPK, and activator protein 1. All this results in the release of downstream mediators like TNF alpha, interleukin 17, and transcription of inflammatory protein and growth factors. We all know interleukin 16, interleukin 6, 13, and TNF alpha will cause inflammation. TH17 is one of the important mediator in autoimmunity, and the transcription of inflammatory mediators is one of the main reasons for the behavioral and cognitive changes seen in the autism spectrum disorders. In case of Parkinson's disease, the dysbiotic gut is found to release a specific protein called curlis. This curlis protein acts as a monomer. It can aggregate together to form a polymer alpha-synuclein fibrils. This on misfolding in the brain, it causes aggregation of amyloid precursor protein, and it will deposit in the substantia nigra in the CNS. We all know the substantia nigra cells are responsible for the dopamine production in the brain neurons. The deposition of this amyloid precursor protein in the substantia nigra decreases the dopamine secretion, so that more motor symptoms is seen in Parkinson's disease. This fibrils also deposit in the gastrointestinal epithelium and disturbs the biofilm formation and causes immune dysregulation and gastrointestinal disturbances. A major portion of people with Parkinson's, they all experience this gastrointestinal disturbances. These fibrils also deposit in the endoplasmic reticulum and the mitochondria, and it causes apoptosis, thereby neuronal loss. There are various studies that can demonstrate. For example, Angelis et al. He demonstrated there is a decrease in anti-inflammatory Faecalibacterium, and there is an increase in Enterobacteriaceae and Sutraliae in autism spectrum disorders patient in mice. Asio et al. Also demonstrated and proved oral supplementation of Bacteria fragilis helps to improve behavioral changes in autism spectrum disorders in mice. Brashfield also reported increase in fumicuteus and decrease in bacteroides in mouse model of Alzheimer's. Oakes et al. Also demonstrated decrease in bacteroid and increase in fumicuteus in Alzheimer's disease patient. Cornetto et al. Showed increase in the number of Shigella bacteria and decrease in Bacteroid fragilis and Eubacterium in a cognitive impaired elderly patients. These are all done in the patient's stool sample. Oral supplementation of probiotics containing Bifidobacterium lactobacillus, Bacillus fragilis can ameliorate the disease condition, especially the cognitive changes and behavioral abnormalities. Thereby it helps in this disease conditions. So there is a need for further research in the field of gut microbiota to identify newer therapeutic agent for the cognitive affected disorder like Alzheimer's disease, autism spectrum disorders, etc. These are my references. Thank you. I once again thank the organizers of Cognitive Neurosciences webinar for giving me this opportunity to present this research. Thank you.